Scales.
Thank you so much. Saad, where are you? That was a very hard act to follow. I'm very happy to be here and to tell you a little bit about some of the work we're doing in my lab. So let's see if this is going to advance. The work that I'm going to show today is the work of Chen Huang. But before I get started on that, I often get ahead of myself and talk too much. So I just want to acknowledge everyone in my lab, also funding agencies, collaborators, and to tell you that we work on a couple of projects. Most of them have to do with understanding the dynamic mapping between structure and function in biological systems at different scales, at different length scales, including fish and animals, different types of animal behavior and subcellular and cellular level. So I was going to start by giving you an outline of what I am not going to talk about today. I got obsessed over the summertime thinking about the evolution of the first mouth in eukaryotic cells and how that's differed completely in the functional design of the cells from bacterial cells to eukaryotic cells. And that was how I spent my summer, basically. But we also work on cilia in the lung, try to understand the emergence of spontaneous coordination and fluid pumping. And we work on locomotion of sea stars and understand how the tube feet, in particular, those muscular hydrostats, how they work to propel the entire sea star body and produce different gates. But for today, I want to focus on collective intelligence or collective behavior, and I want to bring your attention to this school of fish here, taken from some YouTube video where you see thousands and thousands of fish schooling together. And this is another example, bird flocks. This is a very famous example from the work of Kavli and Parisi and others. We know that sheep also, they do this collective motion. This is actually triggered or scared by dogs that are trying to direct the herd in a given direction. And we've heard quite a bit about tissue remodeling and tissue fluidization today earlier. So what do we have new to offer in terms of collective schooling of fish? You probably know about, you all know about the V-check alignment model, where you can produce collective polarization by just allowing particles to basically align in some neighborhood of other particles. And there is a variant of this model where you basically allow for different zones of avoidance, alignment, and attraction, and you can produce three stereotypical behavior at the school level, where you either swarm, so no real order, but you stay in place as a blob. Sorry for using the word blob out of the right context here. Or you could go, the school could go and mill together, so you have rotational order, or you have polarization and the collective movement of the school. So that's all good and really nice, but we are, in these models, we're lacking some ingredients. Here, let's see why. So we don't know how basically all the work that we've been developing as a community on understanding perception action cycles at the individual level, how that translates and connects to collective behavior. So we know that at the individual level, we have some information in the environment or in the actual organism that gets processed by sensory modalities, and that gets maybe transferred to some central nervous system or not, but that produces some action on the environment, and there is the role of the external environment in actually producing the behavior. So there is a lot of work on understanding this cycle on one end, and when we talk about behavior, we have to really specify what type of behavior. In Saad's case, he was talking about the escape of the blob. We have different types of behaviors. Different communities are looking at feeding, predation, evasion, navigation, locomotion, schooling. We kind of dabble in all those different systems or modes of behavior, but for today I'm going to focus on schooling and schooling only. And for my real question that I'm after is how do the emergent collective behaviors that we observe in animal groups reflect what's happening at the individual level in terms of the neuromechanical processes and also the environment, the environmental conditions, the environment that those animals group are living in. So here's an example. I want to tell you that this matters, and I'm going to give you 3 examples to tell you why this matters. These 3 groups of fish, they come from the lab of my longtime collaborator at UC Irvine, Matt McHenry, where they are all relatives. They are all tetras. They have a long Latin name. They are all cousins, but each group schools differently. These are small fish that you would see in an aquarium, or when you go to buy some fish for your kids, you probably would get one of those. They look the same, they look very similar, but they school very differently. So it must be that this emergent behavior is not universal, right? There are no fish school alike, or no types of fish school alike. This is another example. So we want to understand how do those differences emerge, and what's the signature at the group level. Here's another example from a group of researchers at PMMH at USC in Paris. This is Godoy Diana, one of the PIs on this work, where they actually modulated, they put fish in a tank, and they modulated the intensity of the light. And I don't know, this is not working, and this light is not working. But if you look at the dashed line, this is the light intensity, and they increase it, and they decrease it, and they do this periodically. And what they see is that the emergent behavior changes. And we don't know why it's changing. Is it because, thank you so much, let's see if yes. So you go from polarized schooling to milling, you decrease the light, you go to polarized schooling, you increase the light, you go to milling again. And is it that the individuals are changing their behavioral rules? Are the perception action cycles changing, or is just the perception part changing? What's going on here? We don't know really well what's going on. Here's a third example, and this is what I'm going to focus on really, because that's the simplest. These are fish in a tank. This is the type of experimental data we get from Matt McHenry's lab. You see it's in a circular tank. This is another experiment from the group of Ian Cousin, where you do have boundaries. And what they show in this experiment is pretty cool. They show that depending, this is time here, the x-axis is time, depending on the number of fish that they have in their tank, you see this back and forth switching between polarized schooling, all going in one direction, to milling, here, polarized schooling here, and now they are going to go into a milling motion, and then they will interact with the boundary, and then we will switch again. But what's interesting is that those switching rates depend on the number of fish in their tank. So we wanted to understand, that's one of the questions that we were after. Can we understand those emergent phases and phase transitions in confined schools? So we are going to start from a model that was built in Toulouse by Guy Terrola and others, Clément Cyr and others, where they looked at actual fish in a tank, and they were tracking the trajectories, and from this trajectory, it's a data-inferred model, it's experimentally derived model, they would basically come up with a mathematical model for the orientation of each individual as a particle, where you get attracted, an individual gets attracted to its Voronoi neighbors with some intensity. It also aligns with its Voronoi neighbors with another intensity, and it has some rotational noise. And we're going to add an anisotropic visual perception field, and also we are going to modify the work that they did by allowing to have the individual fish has a flow hydrodynamic signature. This is truly in a fluid environment, and we're considering that each fish is going to produce a dipolar flow field around it. Now you might be asking, why dipolar flow field? Is that the real signature of a moving fish? This is based on a PIV data, particle image velocimetry, one of the first to be done on moving fish. And you see that at the front part of the fish, indeed, the flow field is dipolar. But there is a wake and other stuff happening at the tail that we are completely ignoring in the model. So I am not very faithful to the fluid dynamics. I'm capturing a far field effect. This vertical structures would decay with vorticity, so I'm not capturing that in the model. I'm only capturing the dipolar field. All right, so if you are interested in following at this stage of the day, you're probably very tired, but if you're interested in following the dynamical equations, I have particles, self-propelled particles with position and velocity are dot, it's self-propelled velocity plus the velocity of being advected by the flow field created by all the other fish, because every fish is moving the fluid and you feel the net effect of this fluid movement and that moves you with it. And your orientation is changing dynamically as well. There is part of this change is due to a small level of noise. We think of this as the particle or the individual's free will to not follow what it's supposed to do, but it has some free will here, this noise. It has an attraction alignment intensity that's experimentally derived, as I just mentioned to you. With so
attraction and alignment intensity.
Yes, all of it in here. I don't want to write a big expression like this. That's all combined in here. To your Voronoi neighbors, you're only aligning with your immediate Voronoi neighbors, not with everyone. You don't care about the rest of the particles. But then the flow interaction, you are going to reorient with the flow because, of course, the flow field is going to have some gradient at your location, and because you're an elongated body in the flow field, that will affect your orientation. And that flow field is the flow field that's created by all other particles, not just by the immediate Voronoi neighbors. Okay, so I can reduce this model to a few parameters. I have a few parameters here. I have the dipolar intensity, which I'm fixing because I'm assuming they are all the same fish, they are all moving at the same speed, they all have the same dipole intensity. I am only going to vary. I'm also fixing the attraction intensity. I'm normalizing by that. I'm doing a non-dimensionalization that allows me to reduce. The only two parameters that I'm going to vary are the alignment intensity and the noise intensity. The alignment intensity is how much you conform with what your neighbors want you to do. And your noise intensity is your free will of how much you don't give a damn about what your neighbors want you to do. So those are the two parameters that I'm going to vary in the model. And then if this actually does move, okay, so we can now look at what kind of emergent behavior we get.
Could I ask a question?
Of course, please.
Yeah. So the alignment it depends on the nearest Voronoi neighbors. So
yes.
It only cares about the topology of the configuration of the fish and doesn't change with the distance between neighbors?
The Voronoi neighbors will change with distance. Your Voronoi, at every time step, your Voronoi neighbors will be different.
I guess what I mean is that, for instance, if you have a low density of fish,
yes.
then the tessellation would just have larger Voronoi cells.
That's right, and you will be attracted to your Voronoi neighbors. So there is
even if they are far away, that alignment still
yes.
Works.
Okay. Yes.
Doesn't decay with distance.
That's right.
Okay.
That's right. That's right.
It's the same intensity independent as long as they're in the first Voronoi.
Your immediate first
Voronoi.
The same intensity of force? They will have the intensity. Actually, the intensity is the same. It's a constant, but the intensity is multiplying a distance and some orientation. So there are more terms in there. Yes. Very good. Thank you. Okay. So now that you missed my movie, I'm going to have to play it again. Okay. So what do you get with this model? In unbounded domains, you don't have any boundaries. You get actually the three collective phases that you see typically in the three zone model. You get the swarming, you get the schooling, you get the milling. I'm adding the streamlines to show you that I'm accounting for hydrodynamics. But you also get an additional phase. It's this turning phase where the entire school turns. So you have high polarization and you also have high rotational order, and that's what causes you to turn. And we can now, these are results in a previous paper where we considered about 100 fish in unbounded domain, but you can now, of course, scale this to different number, and this is one of the numerical experiments that we can do, and you see the order that is starting to arise in this kind of model. All right, so the question, as I said to you, is how do you behave in confinement? And when we add confinement, and we truly add confinement to basically respect the fluid dynamics, so we have basically a slip boundary condition here at the boundary, but we don't allow fluid to cross to the other side of the domain. And what you see is that you get the 4 stereotypical behaviors that you observe, or that we saw in unbounded domain. You see them again in the bounded domain. You see the swarming, the milling, the schooling, and the turning inside this tank. But and an important feature of this model is that the group is going to swim on average when it's one of those polarized states. It's going to move on average at a higher speed than the individual speed. Individual speed is, the self-propelled speed is constant, but the average school speed is going to be higher because of the fluid dynamics, because we account for hydrodynamic interactions. So that's an important feature of the model. But now when we add confinement, in addition to these 4 phases that we see in unbounded domains, we have additional phases that are there because of confinement. One of them is this following the tank wall. I mean, in most of the experimental data we get from Matt, fish are following the tank wall. Another one is this elongated milling, where the fish go around, turn and come back, go around, turn and come back. This we see in unbounded domain as well. But really this double milling motion, where part of the group goes in one direction and another part of the group goes in the opposite direction, this is a confinement-induced effect as well. Yes?
So when they're following the tank wall,
Yes.
do you have image fish to
I
satisfy
do.
slip boundary conditions?
I do have image fish to allow for the slip boundary condition, but no fluid going. Yes.
Thank you.
Thank you. Yes. All right. So, so we see these new phases, but really as a dynamicist, I think the phase that I am most excited about is this one here. We see this intermittent behavior. Now this is a group of fish inside the tank. Zooming in, it's going around in a milling mode. Looking at the rotational order parameter, it's high. Polarization order parameter is low. But now, all of a sudden, I'm not changing anything in the experiment, the numerical experiment. It's going to transition by itself into this schooling, polarized schooling mode, where now the polarization order is very high and the rotational order is low. And it keeps doing this transition, back and forth transition, from rotational order, from milling to schooling, back to milling, back to schooling. And this is very different than another type of bistability. This is bistable behavior. This is indicating that there are 2 stable modes and you're jumping back and forth between them. This is different than a bistable behavior where you basically go into, at least for the duration of the simulation, you go into either polarized motion or a milling motion, but you don't cross back and forth between them. Okay, so the input to my model are these 2 noise parameter and alignment parameter. For certain, without changing those, I can have intermittent behavior. I change those parameters, I could get 2 different behavior, but no switching between them, at least for the duration of the simulation. Yes?
So in the experiment, how shallow is the water body?
This is all simulation. This is all simulation. And the experiments, it's shallow water experiment. Thank you, thank you. This is the best compliment I get when I show these movies. They look realistic. Thank you so much. All right. So where do we see this behavior in the parameter space, in my parameter space that I described to you? I have a 2-dimensional parameter space, my alignment and noise intensity. I'm going to start with the case when I have no confinement. We reproduce exactly the same data that we had in the PRL 2018 paper where we had no confinement. We have the milling mode, we have the swarming mode, we have the schooling mode, and we have the turning mode or phase. And by the way, each one of these simulations, each one of these dots is a result of 100 different Monte Carlo simulations for this parameter value. So this is a very expensive parameter space to construct. Now, if I add the confinement, the circular boundary, but I don't, because I'm doing simulations, I can remove the fluid completely, and I can see what do I get by adding only the boundary effect with no fluid. And you see that this is going to lead to this following the tank wall domain phase that emerges here. And I also have the bistability and the intermittent bistability. The two, this is a confinement-induced effect. And if I have both the fluid, the hydrodynamic interaction, and the confinement, I get back the turning phase, because the turning phase is really dependent on hydrodynamic interaction. It doesn't exist without the hydrodynamic interaction. It's a flow-induced phase. All right, so these three phases are due to confinement. You have something else you wanted to say? It's a kinematic model. All the model is kinematic. The flow signature is dipolar. It's basically point dipoles interacting together, right? But you have to account for all dipolar interaction. All right, now if I reduce the number of individuals in this tank from 100 to 10, I'm going to see these new exotic modes that I showed you, this double milling, which I think is very interesting to have this group split into two groups, one going in one direction, the other going in the opposite direction. And I kind of have a theory of, or maybe a conjecture of why this could be useful. This could be useful potentially for mixing oxygen or mixing in an actual fish. I tried to see if there are any reports experimentally of fish doing this in a tank. The only thing I found is that there are fish farms where you have to grow a lot of fish, or farming fish in tanks, very dense, and then they go into opposite directions. So that's the closest I got to finding the double milling behavior experimentally. Okay, so how are these modes dependent on the density? What controls this transition? I told you I kept the domain size the same, but I reduced the number of fish. I have now new emergent behavior. So I wanted to see if density is the control parameter or if the behavior really depends on the domain size and the number of individuals. And this is a parameter space where I have the confinement radius, basically the tank radius, and the school size. And the black lines are constant density. The density is constant. And you see that even as you go at the same contour line of density, you keep the density constant, you see a transition from milling to an elongated milling phase. So that means that density alone is not the control parameter. That's unlike the V-check model, where you only control the density. And we can look at what we get out of the model. We can compare to experimental data that were reported in the cousin paper, where they did this experiment. They actually maintained the same density by reducing the tank size and reducing proportionally the number of fish. And they saw a similar transition from high rotational order to something where you don't see much order. So this element is, again, captured in the model. All right, so now to look at the element that was reported in those experiments where you saw the transition between polarization and rotational order, in our model as well, you see this transition between polarization and rotational order, and you see that this transition or the switching rate changes with the number of individuals in the school. And this is our model results, so the amount of time you spend in one of the states, I think at the schooling state is red, and you see how the school size affects the amount of time you spend in each state in the model versus in the experiment, and qualitatively they have the same trend. with more fish, you spend more time in one state. All right, now let's talk about how can we analyze this collective behavior. 5 minutes, very good, okay, excellent. So let's see if we can analyze this. We can actually look at this intermittent behavior. This is the dynamic transitions, basically. And this bistable behavior where depending on your initial conditions, you can get into one state or another. But if you look at histograms over Monte Carlo simulations of these two different scenarios, you get the same, you see the same thing. You see basically a bimodal distribution. So looking at histograms is not going to give you any information about the dynamics. And this is not because of limitations of resolution or accuracy. This is really fundamental in how you build a histogram based on those realizations, Monte Carlo realizations, where you can go to one state or another state. You wouldn't know if this allows for transitions between states or it doesn't by looking at histograms. So we needed a method, a dynamic analysis method that allows us to see if we can transition between these two states. And the way we did that is by lifting the dynamics from individual level dynamics to group level dynamics on the coarse-grained polarization and rotational order parameters. We think of a Fokker-Planck equation that is defined on this lifted space, but luckily our data collapses onto a one-dimensional subspace in that lifted space. It doesn't even live on the entire p-pi space. It lives on a nonlinear kernel, which we can project and get to that nonlinear kernel and find the effective potential of the Fokker-Planck described on this one-dimensional subspace. And we can now look at, we can construct this effective potential, and we can see that in the intermittent state, we have a finite potential barrier. When we go into those bistable states but no switching between them, we have an infinite potential barrier between the two states. And we can do something similar to what we do in dynamical systems analysis, where we change the parameter value. We look at the where are those collective equilibria, if you like, or collective phases, and how you transition from bistability to intermittency as you increase the noise intensity and eventually to only schooling, where you start from an infinite barrier, you go to a finite barrier, and then you have a bifurcation that looks like a saddle node bifurcation where you end up with schooling only. You can change the school size and do the same again. And because I'm running out of time, I just wanted to show you what happens at extreme scales when we now go from 100 fish to 1,000 to 10,000. You don't get the same behavior at all, right? There is a place where your behavior, when you have 10,000 individuals, is very different. The school is going to organize, at least try to organize, and then it's going to continuously split and merge when you're supposed to be in a schooling state. When you're supposed to be in a milling state, you see something similar to what you would call active turbulence. And I just wanted to show you this simulation with 20,000 fish. What you are looking at here, the color is only to allow you to basically keep track of those individuals and to see if they will mix together as the school evolves. And this is what you're going to see. The colors start to mix immediately. You have neighbors exchanging with other neighbors. You have density fronts that are developing. You have rarefication waves that are developing. So all of this exciting dynamics, that's actually quite difficult to analyze. So I'm going to skip what we're doing on understanding sensory cues. I just wanted to show you this because this is a group of physicists. And I mean, come on, this is like Joule's experiment with the bichamber fish and a bichamber domain with a little channel between. And I want to tell you, if you have a single fish, it's never going to escape from one channel to another. Now you're looking at a few, I don't know, maybe 100 here, and you are going to see what's going to happen. Okay, first approach, no escape. Do you think they are going to escape? I mean, this is just fresh off my student desk. That's why it's very slow movie. But here we go. Now they escape to the other side. This is a group. This is really a collective effect. We analyzed the single fish. It does not escape at all. And we have reasons to believe why, to know why. But now they are going to transition back and forth and continue to transition between the two chambers. It's not a symmetric effect. All right. So I want to thank you, and I'm happy to take any questions and go to dinner.
Maybe I'll start with one very fast while. So the milling that you're observing, so the old Ian Cousins model had this criticism that when you observe milling, it's only because your attraction is of the size of your mill, essentially, your attraction range. And the one that you're observing, is it because you have the cone of vision, or is it because you also have long-range attraction?
I don't know the exact answer to this. I didn't try to have the visual field to remove the blind spot in the back.
But your attraction length is what scale?
Length is any scale,
is any,
as we said.
yeah, okay, so
right?
it could span the...
It could, yes.
Okay,
So,
right. Go ahead.
Very cool talk. So I had a question suggested by an experiment that my friend Amit Kele was doing a few years ago. So he was having a few of these toy fish, you know, that you can put in the bathroom for kids with the tail, essentially dipoles. And then he was putting them in confinement and studied how they would behave. But he had two kinds of confinement. One was walls, and the other one was a mesh of the same size within the larger pool. And of course, the boundary conditions for the hydrodynamic flows are completely different. And then it's on your confinement for the fish. And he observed very, very different behavior. So I was wondering whether amongst the things that you tried, you could also try that and see if it conditioned different kind of collective dynamics.
So what do you mean by...
The idea is that if you have a mesh, well, then you don't have the image fish.
Yes.
The mesh is transparent to the fluid, but not to the fish.
That's right.
And then what happens in that scenario? So you have a hard body constraint, but not for the flow.
Right.
So that was more like a suggestion.
Right. That's going to be about steric, because I don't know if the fish could even see that mesh. Because here...
Well, you could imagine he sees. You could decide that he can see the mesh, and so then you can encode it.
In the bichamber domain, the hurdle is that you don't see because you have a solid obstruction. If we now were to remove the solid obstruction and allow just by steric, you have to really hit against the mesh in order to know it's there. Then it becomes a different mechanism. Yeah. But it will be very interesting to look into that. Thank you for the suggestion.
I guess we'll switch since there's so many people on both sides.
Okay.
I had a question on your intermittency versus bistability.
Yes.
Do you think that's really a qualitative distinction, or is it just a matter of time scale?
I don't know. I know about, all I can say is about the time of my simulation. I can't tell anything about what happens after my simulation time.
Sure, but in your phase diagram, as you approach that sort of phase boundary, does the time scale of the intermittent switching grow?
Yes, I did show the rate of switching. You see, I did a rigorous, as much as the data can allow me, dynamical systems analysis, and I see that this switching rate is increasing as I approach as the two stable states don't exist anymore. Yes.
Okay, thanks.
Good point.
You know, normally when one talks about a phase, one is somehow trying to understand the limit of infinite size at fixed density. Is it that these systems, it's pointless trying to ask about what is the phase in the infinite size limit, or is it that these are systems whose generic state is kind of phase separated into high density and low density? So if you just...
I'm humbled that you're asking me this question. This is a question I wanted to ask you because I'm trying the simulation. In the infinite, I'm trying simulations with 10, 20, 50,000 fish, and I see that beyond a certain number, I cannot talk about any emergent, cohesive, collective order. I mean, I do have other interesting dynamics that emerges. So yes, I would love to have an answer.
One mini question.
Yes.
the transitions you saw at finite n, at very small n, are those a result of what is called demographic noise, where you have a noise in the finite n? You write down equations of motion for order parameters for a system of a finite number, and then the noise in those coarse-grained but finitely coarse-grained equations have dependencies roughly 1 over root n or 1 over root n times 1 minus n or something like that.
The noise doesn't, but yes, the noise doesn't depend.
No, the effective noise for the corrective variable will depend on n.
Sure,
and is it perhaps a result of that? Are those transitions a result?
So all I can say is if I hold the noise fixed and I don't consider it in my parameter space, I do a parameter space on attraction and alignment intensity at a fixed noise, I still get
that's when as a function of n, you see some behaviors when you change n.
Yes.
And so that is like because of
that would be the effective noise, yes. But I'd love to talk about those things. Yes.
Go ahead.
Hi. Hi.
Sorry. Yes.
Excellent talk. My question was kind of related to that, but it was more like in the sense of more is different, right? So usually the way we think about it is like there's new interactions that are introduced at larger scale. So introduce more and more complexity that leads to this. So in the sense that the energy flow is more like a monodromous, right? You can go down, you cannot go up. But in your case, the interactions do not change, just the number of fish.
Yes.
But
there is an effective noise, and there is also the effective hydrodynamic interaction.
Okay, right? I understand, but these are all there throughout the problem, right?
Yes.
So do you hit some critical point where?
Yes, I do. I do.
I see.
I mean, I thought I showed this, but I didn't emphasize it much.
Well,
this is what's happening, right? Okay, go back. When I am at this, when I'm changing the number of fish systematically, and I'm starting at a schooling where the group is supposed to school with high polarization, then it decays after a certain number. You start seeing some fluctuation and the gradual decay. In the milling case, it looks like a phase transition to me.
Okay, it looks more. I see. Okay, I saw that deep. It's okay.
I don't know. What's going on? I don't know. I mean, I
think
at
some point it started to jump.
With two different.
Okay, thanks. We can chat later. I don't want to take all the.
Yeah, I think the question was answered.
Yes,
so
I think the question was
maybe we
answered,
can go to the other side. The other side again. Yeah, switching all the time.
Of course. I can't. It's my intermittent behavior.
Thank you very much.
Yes.
Regarding your observation about the colored areas that mix, so basically individual change their neighbors.
Yes.
And I'm not an expert of bird flocking, but from popular science things, I seem to remember that this is not the case for birds. So if that's the case, what's the difference?
My story is longer, a little bit. When I am in a polarized state, even with the fish, even with the model, this is a model fish, it could be anything. I could call it birds, and you might believe me. But when I am in the schooling phase, when I expect polarization, and then I do see this splitting and splitting and merging with higher numbers, the merging you don't switch your neighbors when you're in a polarized state. And in Cavagna's work, all of it is on polarized states. There is no analysis of this milling state. Now, when I'm in the milling state, it's a much different scenario. Propagation of information is not only because your neighbor is affected by what you do and your neighbor is affected by what your neighbor did. It's actually because you're changing your neighbors. So that's only in the milling case. Does that make sense what I'm trying to say? In the schooling, in the polarization case, we see exactly the same results of Cavagna. We can reproduce all of it. In the milling case, we don't see because Cavagna didn't work on the milling case. We see new behavior that we don't see there.
Thank you very much.
Thank you. Thank you for the question. Yes.
Yeah, so two very small questions. But when you see the intermittency, and it's when you have confinement, is there some relation between when you switch and how close you are to the boundary?
Good question. Excellent question. very faint relationship, because you can change. So there is a parameter that I didn't mention to you, which is like when you see the boundary, you actually turn, right? And this parameter could be very high, so you turn very far away from the boundary, or you could wait until you're very close to the boundary and turn. And in both cases, we see intermittency. So this parameter didn't have much effect. It had some quantitative effects, but not a qualitative effect. That's why I ignored it completely.
Okay, and the other thing is, have you thought of, instead of confinement, introducing some obstacle in the middle or something?
Yes, excellent question. Yes, I was more interested in the bi-chamber one, but yes, I was hoping that there would be a hierarchy of stuff. Depending on the patience of the graduate students, Saad said they would just pack up and leave. I don't know what my graduate students would do, but yes, we're trying. You see, just constructing the boundary numerically is a feat by itself. The non-regular boundary, because when it's a circular boundary only, like David asked about image system, it's simple. You use an image system. When you have a non-regular boundary, you have to be more careful in constructing that boundary to account for the hydrodynamics properly.
Yeah, very nice talk. So you had started by saying that you want to understand the perception action cycles. And so what would you say the fish are feeling, seeing this interesting changes in system size?
Thank you for the question.
Are they seeing like,
yes.
yeah.
Thank you for the question. I love it. So what I'm saying is that there are transitions that happen when mathematically I have the same action perception cycle and the same parameter value. I'm not changing the parameter value and I see those transitions, this intermittency. So this means to me that it is possible to have transition that's driven collectively by the group dynamics, and it's not because of an individual change or a change at the individual level, because it's very hard to tell that from an experiment, but it's easy to make that assertion in a model because I know what went into the individual level dynamics. So I can see with the same individual level dynamics, I see a group level action that causes this change in behavior. But I also see, like I showed you, even when you change the individual level dynamics by changing those parameters, you also have transitions, which is not surprising because you would expect that if you change the behavior at the individual level, you should see that reflected at the collective level. The more surprising case is when you have no change at the individual level, but you have change at the collective level. Thank you for the question. Yes. This way?
I guess
Okay, good.
I guess you saw that it was
I guess you're changing the rules, you're confusing.
So thanks for a very beautiful talk. You have essentially a system of particles that are also interacting via a flow field.
Yes.
So you showed many beautiful phases of on the particle side of things.
Yes.
Are there also, presumably there are correspondingly interesting things happening also with the flow, like maybe funny dynamics of topological defects or something like that?
Yes, very good question. We're starting to look at that in the bi-chamber case because we know when we have only a single individual, the probability of escaping is almost zero for the duration of our integration time. And when you have multiple, the flow is going to help to escape. So it's a collective effect and flow is playing a role in there. And that's when we started paying attention to the flow field. But beyond just showing you the streamlines, I showed you some streamlines. Some of them look very ordered, so the entire school has a signature. You can start asking like what's the hydrodynamic signature of the collective mode? And the swarming mode, the hydrodynamic signature is more turbulent. In the ordered mode, the entire school generates a dipole. So yes, there is something there to explore. Thank you for the question.
Last question.
I once heard that schools and flocks and so on help animals evade predators. Is that true? And can you model that either in experiment with others or here in simulation?
We have a couple of preliminary simulations with a predator coming in, a dumb predator because it doesn't change its direction, and the entire school separates and reassembles. So yes, we're thinking along those lines. Yes.
But then to settle the Wikipedia trivia pub question, is it true though that schools help with avoiding predators?
Okay, this is not philosophy, this is not science. I think for every predation strategy, there is an evasion strategy. And for every evasion strategy, a predator evolves to take advantage of it. So this is, sorry? It's an arms race. Every time you develop an evasion strategy, a predator will come and take it.
So I can make a quick comment. I worked a lot in this field several years ago, and if you ask biologists, they don't agree, typically, but there's many reasons why they move collectively. One of them is argued for predators, but in different species, they say like it's, in one case, it's to protect the ones in the middle, in the other case, it's to strong, and there's weak correlations, and it depends on the strategy. So it seems to be part of the strategy. Most people are thinking a lot that the collective intelligence aspect is it. I mean, a lot of people are thinking that the collective intelligence is quite important because collectively they seem to be able to do things that they can't individually, like finding food more reliable and stuff like that. But the ultimate reasons, you know, you ask 3 biologists and you get 5 reasons, right? So about why they move collectively. So there's probably more than one reason.
Engineers think it's for hydrodynamic
right,
advantage.
Exactly. That's another reason.
We do a different type of models with CFD type models, and we see that you have locations in a group that are hydrodynamically advantageous, but there are locations that are very bad for you. And there are formations that produce a wake that doesn't allow anyone to come in. So kind of a selfish formation. You can talk about, and you can talk about the groups or individuals within a group changing their locations within the group because they are competing for the most advantageous location energetically. I mean, all of these are hypotheses, but we can see those locations in the hydrodynamics, that there are some that offer benefits to a follower while others don't offer any.
Sounds like a peloton in cycling.
Yeah,
cool, thanks.
Yes,
absolutely. All right, with that,
all right.
we'll thank
you so
Eva
much.
again and
Thank you.
go for dinner.